in different methods of reproductions. This is about the vegetative propagation, and next coming to the asexual reproduction. Asexual reproduction is also single parent reproduction. That is, here only one parent is participating in the reproduction, and here as like that of vegetative propagation, here also single parent is taking part, and whatever the characters that are there in the parent plant, the same type of characters will be retained in the daughter generation of plants also. Daughter generation of plant, and whatever the genetic constitution or whatever the good characters or whatever the characters whichever that are present in the parent, the same will be retained in the offspring generation also. And there is no possibility of incorporating the new characters, and there is no possibility of variations, and there is no possibility of incorporating the new genetical variations into the offsprings. So that there are some advantages and disadvantages related to the asexual reproduction, and there are various methods of asexual reproductions taking place in the plants. And here, before going to the types of asexual reproductions, there are different structures or different special type of structures which are produced during the asexual reproduction. Those structures are called as spores. And no gamete formation. No fusion of gametes. No meiosis. Only the mitotic type of division is taking place, because when the gametes are fused, it results in zygote in the sexual reproduction where there is a need of meiosis. But here, as there is no fusion of gametes, so the spores which have been formed, so whatever the spores or whatever the asexual propagules or asexual structures, so the ploidy of the asexual structures will be same as like that of the parent generation. So those structures will undergo repeated mitosis to produce the similar type of parent where the ploidy will also be retained same. So that these are some of the characters related to the asexual reproduction. And when we are talking about the spores, there are two types of spores are there. One is some are non-motile spores, and some of them are motile spores also. So let us discuss some of the types of asexual reproductions which are taking place in the different groups of living organisms. Okay. There are many methods of asexual reproductions. So let us discuss some of the types of asexual reproductions here. One is binary fission. Fission means division of the parent organism into two equal halves, so that each of the half is developing into a daughter plants or daughter organisms. This type of binary fission is most commonly observed in bacteria, amoeba, paramecium, euglena like organisms. So here let us take the example, and one more thing, all of them are unicellular organisms. As we know that all the living organisms are having death at one or other stage of their life, but here in the unicellular organisms, like such examples, so there is no natural death for these organisms, such that there is no natural death means here at one point of time when they participate in the binary fission, the parent organism, as we are saying that the parent organism is made up of only one cell, that one cell of the parent organism will divide into two equal halves, and each of the cell which is resulted here, that will be growing into a daughter generation or daughter organism, so that the life of the parent is continued in the form of daughter generations. So there is no natural death for these unicellular organisms. So let us take the example of bacteria here. So here bacteria is a unicellular organism which contains cell wall and plasma membrane. Inside the plasma membrane, the nuclear material, that is the nucleoid we call, because it is a prokaryotic organism, along with they contains the different types of cell constituents. First, a small constriction will develop in the middle of the cell that is on the outer side. So, this constriction that is developing on the middle will gradually grow towards the center. In the whatever the constriction that have been formed will gradually grow into the cytoplasm, and this constriction, when it is moving towards the center, both the constrictions will move towards the center and they will unite at one part, and here the constriction is moving from periphery to the center, so that is called as a centripetal manner. The constriction is moving in a centripetal manner, and after some time, so before that what happens, the nucleus will also be divided into two daughter nuclei here. Then the constriction will start in the cell wall also. This constriction which have been formed in the cell wall at the same place where the constriction have been occurred in the cytoplasmic contents. So, it also move towards the center, and finally it will be resulting into two daughter bacteria. So, this is a process of binary fission where the parent cell is divided by normal process to form into two daughter cells, and two daughter cells are similar to the parent and each of them are similar to each other, and this type of fission is called as a binary fission. And this type of binary fission can be observed in examples like bacteria, amoeba, paramecium and euglena. In bacteria, this is a method where we call it as a transverse binary fission, whereas in amoeba the simple binary fission is present. In paramecium, transverse binary fission is present, whereas in euglena the longitudinal binary fission is there. So, in longitudinal binary fission, first the flagella will divide into two flagella, later the body of the euglena will also be divided longitudinally to form into two daughter euglena parts. Fragmentation. In some plants, starting from lower to the higher plants, so here what happens is the plant body is broken into small fragments. Let us take the example of Spirogyra, which is an algal member, and in Rhizopus, that is a fungal member. Rixia, that is in bryophyte. In some of these examples, the plant body is broken into small fragments, and each of the fragment will develop into a daughter individual independent organism. So, this type of small pieces which have been separated and developing into small individual daughter plants, so we call this process fragmentation process. Next one is sporulation. Sporulation is a process where the plant will produce a small special type of structures called as spores, and these spores are generally, in most of the cases, these are unicellular, and the ploidy will be generally haploid, or sometimes they may be diploid also, but in most of the cases they will be haploid, and they are having a small layer around them. These are spherical in their nature, and these spores may be either motile. If they are motile, we call them as zoospores, and if they are non-motile, we call them as aplanospores. Zoospores, when how does they act as a motile spores means they have some locomotive structures called as either flagella or cilia. With the help of the flagella or cilia, they will swim in the water, and they will reach the suitable substratum and germinate to give rise to the daughter generation of organisms. Whereas in the non-motile spores, as like that is aplanospores, they are passively dispersed with the help of wind or any external agent, and they fall on suitable substratum and germinate to give rise to the daughter plants or daughter organisms. different types of spores are formed by different groups of plant species. Let us take some of the examples and the types of spores that have been formed by various types of plant species. The first one is zoospores. Zoospores, as we have said, that these are the motile spores, and these are produced inside a special structure called as zoosporangium, or plural we call it as zoosporangia. And these zoosporangiums, this can be seen in Chlamydomonas example, algae. In which the parent Chlamydomonas is an unicellular organism and aquatic organism. So here the cytoplasmic contents of this Chlamydomonas is divided into similar type of structures called as spores, and each of the spores will contains flagella. So when the parent Chlamydomonas is broken, the daughter spores will be released outside. These are the similar type of spores which have been formed by the Chlamydomonas species. So these are the zoospores which are formed by Chlamydomonas. So these zoospores will swim with the help of the flagella in the water, and they grow into an individual daughter plants or daughter Chlamydomonas organism. Next one is oidea. Oidea are the special type of spores where that can be seen in Agaricus species. These are simple structures where they does not store food materials. So if you observe the structure, so the they are separated into small fragments like this. So here, these are the oidea, or the small unicellular structures which have been separated from the parent, and each of the fragment will develop into a daughter structure. So this can be seen in Agaricus species. Next one is chlamydospores. Chlamydospores are generally non-motile spores as like oidea, but the difference is they store food materials. These are also the non-motile spores as like that of oidea, but they store the food materials, and these are either formed in intercalary. Intercalary means they are formed in between the cells or at the apices, at the tips also. So when they are formed in the intercalary manner, and one more thing, they will have a thick cell wall around them, thick cell wall, such that they have to retain in unfavorable conditions. So here if you observe the structure of this chlamydospores, so they have some thick structures like this, and this is a diagram of intercalary chlamydospores. These are the chlamydospores. So because the chlamydospores is flanked on either side of the either side by the normal cells, that's why we call it as an intercalary position. Sometimes they may be formed at the tips of the hypha also. This type of chlamydospores can be seen in Rhizopus. Next one is sporangiospores. Sporangiospores are observed in Rhizopus species, which are formed during the favorable conditions. Actually here the sporangiospores are formed inside a stalk like structure. The stalk of this sporangium we can call. So here the stalk like structure is called as sporangiophore. The tip of the sporangiophore become bulged, so this is called as sporangium, and inside the sporangium the spores are present. These spores are called as sporangiospores. The sporangiospores are haploid structures which either may contains one nucleus or sometimes more than one nucleus also, and when the sporangial wall is broken all the sporangiospores are liberated out, and these are the non-motile spores. They are such that they are carried by the wind, and when they fall on suitable substratum, each of the sporangiospore will germinate and give rise to the daughter plant. So this can be seen in Rhizopus species. Here, as the sporangiospores are formed inside the sporangium, so these are endogenous in their origin. Endogenous in their origin. Okay, next one is conidia. Conidia are the spores which are formed in penicillium. Here, these are exogenously formed. That is, they are formed outside. So these are called as exogenous in their origin. And here they have the stalk-like structure called as conidiophore. This is the conidiophore. On the conidiophore, we can see the formation of conidia like this, which are formed outside. So we call the conidia as exogenous in their nature. These are also non-motile spores, and when they are liberated, carried by passive winds, and they will fall on suitable substratum and germinate to give rise to the daughter plants. So these are the 5 types of the spores which are observed in various groups of plants. The next type of asexual reproduction is by gemmae. Gemmae are the special type of asexual structures which are observed in bryophytes like Marchantia. Marchantia is an dioecious plant, that is male prothallus is separate and the female prothallus is separate, and on the prothallus, consider this as a prothallus. So on the lower side of the prothallus, the rhizoids are present. Then on the lower side of this one, we can observe some receptacle like structures. These are called as receptacles, or we can call them gemma cups. Inside the receptacles, multicellular green haploid structures are formed. These multicellular green haploid structures are called as gemma or gemmae. These gemma, each of the gemma will germinate and give rise to the daughter generation. So this can be observed in bryophytes like in Marchantia. Next one is budding. In binary fission, the division of the parent cell is into equal parts, but here the division is there in budding also, but the division is unequal division. That is, the parent will divide to form small protuberance, and that protuberance is called as a bud, and that bud will separate out from the parent plant or parent organism and develop into a new daughter plant. So here this budding can be observed in yeast. Yeast is an unicellular fungi. If we observe the structure, it appears to be like this, which contains nucleus, vacuolar organization, and the protoplasmic content here. Okay. So here first the nucleus will elongate, and by the meantime the cytoplasmic content will produce a small protuberance on one side. The nucleus will completely divide to form into two daughter nuclei. One will be retained in the parent cell, and other will enter into the small protuberance that have been formed by the parent yeast organism. And this structure is called as bud. Sometimes a single bud may be formed on the parent yeast, or sometimes many number of buds may be formed, and each of the bud will separate from the parent and develop into daughter organism. But sometimes what happens, each bud will produce many number of small buds on it. So each of the bud will produce the daughter buds. when such daughter buds are formed, it appears to be like a stage, it appears to be like an organism called as torula. There is a other yeast called as torula. So as the structure is appearing like a torula, so we call this process as torulation, and this as a torula stage. This can be observed in yeast during the budding process. So here the buds are formed because of unequal division of the parent, so that a small protuberance is separated out and developing into a small bud, giving rise to the daughter organism.